Now that we've talked about units of radiation dose and the short and long-term effects of radiation on biological tissues, from the cellular level to the organism level, I'll explain to you how we use the damaging power of radiation to our benefit to help cure cancer by killing the tumor cells while leaving the others unharmed, or by imaging or identifying where the tumor cells could be. The basic premise of all these types of therapy is to target the cancer cells with radiation and leave the surrounding ones as unharmed as possible. And there are 5 main types that are used today: X-ray therapy, brachytherapy, tagging or imaging, proton therapy, and boron-neutron capture therapy, or BNCT, that we briefly introduced earlier in this module. Let's start with X-ray therapy, going all the way back to the beginning of our module. This is based on the differential absorption or attenuation of X-rays in different bits of tissue. On the left, you can see the original X-ray as taken by Röntgen, and on the right, you can see a much more modern X-ray, where we've gotten much better at distinguishing the different amounts of attenuation and absorption of X-rays as they pass through different types of tissue. Well, we use this different amount of absorption to our advantage. Shown here is how X-ray therapy is performed today. Let's say you had a cancer that's completely inoperable, cancer at the base of the brainstem, for example. You may want to target that tissue with radiation to kill those tumor cells, but you don't necessarily want to blast the surrounding brain tissue, or that defeats the purpose of the treatment. So by taking narrowly focused X-ray beams and coming in at this tumor from many different angles, you can make sure that the tumor receives the full amount of X-ray radiation, while each bit of the surrounding tissue only gets a little bit. And this is one way of minimizing damage to the surrounding tissue while maximizing damage to the tumor itself. However, we have a problem in that photons simply travel through matter, so a lot of the rest of the brain, or whatever you're targeting, still gets irradiated. So instead, we can actually use an accelerator or cyclotron to accelerate protons and fire them into the tumor. In this case, these are smallish accelerators. They're small enough to be located at hospitals, like Mass General Hospital down the road, and they function with the principle of cyclotrons, where a positively charged ion, in this case a proton, is injected inside the center of 2 D-shaped electromagnets, called Ds for short, and it's accelerated as it travels in the gap faster and faster and faster, spinning further and further, until they're ejected out the side with a very targeted energy and a very targeted position. They're then sent through bending magnets and collimators to send that proton beam into the tissue of choice. Shown here is a picture of one, whereas before I showed you what one of these proton therapy gantries actually looked like. And this makes possible what's called intensity modulated radiation therapy, or IMRT for short. In this case, the tumor is imaged and modeled, and you can tune the energy and direction of each of these protons so that they actually travel through the tissue, and they do their most damage where they stop, right in the tumor, sparing the tissue on the other side of the tumor from much more radiation. There's still a little bit of radiation generated, but it's a lot less harmful than X-ray radiation. And so now I'd actually like to show you a little bit why. If we look at this graph here, we're actually graphing the relative amount of damage done by X-rays as well as protons. So let's zoom in a little bit. The blue curves show you the amount of damage done as a function of distance by protons as they travel through material. And the stacked blue curves right there show you different amounts of damage versus distance as a function of proton energy, resulting in the red curve, what's seen as SOBP, which shows you that you can irradiate the tumor as much as possible while minimizing dose before and especially after the range of the protons. If we compare that proton dose to X-ray dose, we can see how these 2 very different types of radiation yield drastically different results in radiation damage versus distance to the tissue. Shown in blue is the proton beam, which does a little bit of damage on its way into the tumor, tons of damage within the tumor, and then the protons stop and there's no more damage. Whereas X-rays exponentially attenuate. They do most of their damage as they enter the tissue, a little bit in the tumor, and then a little bit less, but not zero, on the other side of the tumor, which shows you how much more effective and targeted proton therapy can be compared to X-ray therapy. It does require a lot more specialization and a lot more specialized equipment, which is why it's less popular, but its popularity is growing as more and more places gain this capability. There's also another type called brachytherapy, which relies on these little seeds shown here in the picture of a radioactive isotope, like iridium-192, whose radioactive decay can be very well known, and therefore the radiation dose and distance can be very well controlled. The one shown here, iridium-92, relies on beta decay, where most of the time radioactive decay proceeds by beta radiation, and these beta particles have a range of only a few hundred microns to millimeters in healthy tissue and in tumor tissue. So what you can do is actually implant these brachytherapy seeds, usually through a convenient access port in the human body, or somewhere very nearby, where you can use ultrasound imaging, in this case in the convenient rear access port of the human, while you implant lots of little brachytherapy seeds to only locally irradiate the surrounding tissue while leaving the surrounding area outside, in this case the prostate, mostly untouched. You can also use the power of radiation to perform medical imaging, in this case using molybdenum-99 and its beta decay to technetium-99 metastable, that's what the M stands for, and the eventual slower decay of that technetium-99 metastable to the ground state of technetium-99. What it relies on is the very fast beta decay of molybdenum-99 to any number of excited energy states of technetium-99. And then they all hang out there for a little while, and with about a 6-day half-life, which is extraordinarily long for a metastable state, that technetium-99m decays via very well-known gamma-ray energy to technetium-99 at the ground state. And we can tune energy and position-dependent detectors to figure out where in the body the technetium-99 goes. So you can take this technetium-99 isotope, attach it to a biomolecule or a marker, which will grab onto the tumor or the other tissue that you want to image, and then it'll light up on a gamma imaging screen. This is also one of the reasons why if you go to an airport shortly after a medical imaging procedure, you should tell security what you've done, because you will be emitting this radiation which was used to help image and find the tumor, but it also might scare people at the airport. Finally, there's a rather advanced form of radiation therapy called boron-neutron capture therapy. This relies on an accelerator or a nuclear reactor to fire protons into the patient sitting on a gantry. Now let's explain in detail how this works. In this case, a proton accelerator fires into a beryllium target, releasing neutrons, awfully similar to Chadwick's 1932 experiment. Now, through a series of cadmium and lead filters, the neutrons are slowed down to what's called epithermal energies, or roughly 1 to 1,000 electron volts, or eV, such that those neutrons slow down so that their cross-section, or their probability of interaction, gets higher and higher. Now, this patient has been infused with a boron-containing compound that attaches very well to the tumor cells without attaching to the normal cells. Therefore, when the neutrons enter the brain or the other inoperable tumor, they pass through the healthy living tissue, and when they encounter one of the boron-tagged cancer cells, they destroy those cells much more frequently. In this way, you have a very high kill rate of tumor cells while leaving most of the rest of the body or the brain completely untouched. And this was actually tested and demonstrated in a few places around the world, like Finland, Japan, even here at the MIT nuclear reactor. So at our reactor, we actually have a core which produces lots of neutrons and gamma rays, a gamma shield, which you can see what's called the photon shield, and something to select only the fast neutrons, in this case what's called a cadmium fast flux trap, and then a series of lead filters and water moderators to help slow down these neutrons and focus them into a tight epithermal beam, where on the right the cancer patient would sit, and with a very well placed positioning, the neutrons would irradiate the tumor that's already been tagged with the boron, while leaving the rest of the patient mostly unharmed. It actually underwent clinical trials even here at the MIT reactor, but the program was stopped here due to, well, it's costly. Reactor's an awfully costly thing to operate. However, there are now accelerator-based versions that are undergoing clinical testing as we speak, and there are a couple BNCT projects around the world which we hope will get to actual treatment because of its effectiveness.